Today, we are interviewing the Ph.D. student Sammi Surber, who is working in Hsieh lab at the University of Georgia. Let's meet Sammi.
It's my pleasure to have you here. So, would you please tell me something about your background and how brought you from biochemistry to plant biology?
Yeah, so my background, I'm from Nebraska. I grew up on a farm. My whole family are nurses. I am not, don't do well in the medical field. Learned that at a young age. I didn't enjoy that. So growing up on a farm, it was really like what exposed me to being in plant life and kind of like wondering what's going on around me a lot. I loved being outside and I loved playing in the dirt and all of those things. And so like it was just kind of a natural thing where I was like, I like science, I like plants, so I'm just like, I'm gonna do biochemistry. It's really broad, I can do a lot of things with it. And I can learn a lot, and if I really do decide I want to be a doctor or something, I can do that. And in college it just like really solidified that I didn't want to do that type of work. And then I got really connected with plant people at Nebraska, at the University of Nebraska. And I worked in a different lab when I was an underclassman, and I just really wasn't enjoying it. Wasn't liking science at that time, but I just don't think I jived with my mentor at that time. And so then I took a random botany class, and the professor and I just got along really well. I could talk to him about things and like about what I wanted to do in the future. And he got me connected with my advisor as an undergrad, and she really helped me realize how cool plants were. And there's so much more to them than just photosynthesis. So I learned a lot about lipids. I still love lipids. I'm glad CJ's letting me do a side project with them, even though it's been really slow. But I really am not good at chemistry. So I ended up really evaluating what I enjoyed doing, and that was doing molecular work. And I just decided to stay in plant biology instead of doing like micro or something like that. So that's why I moved away from chemistry. It's just because I was not super great at it and was just trying to utilize my benefits.
You mentioned that you are working on lipid as a side project. How about your main project?
Yeah, so my main project is working on sulfate transport in poplar trees right now. And I have 2 gene targets, and they're in a gene family that aren't categorized really in any species, not even Arabidopsis. So there's a lot of confusion onto what they're doing, what their purpose is, if the category is even what we think it is, and those things. So my project is really trying to figure out what their role is, especially in woody species, because it's so different than Arabidopsis, and especially in their xylem and vascular structure, where my genes are highly expressed, so there's just a lot of disconnect in those. So my overall main goal would be looking and characterizing those genes and those transporters as proteins.
Yeah, would it be beneficial to like bioenergy crop development?
Yeah, so obviously being in CJ's lab, we always want to tie it back to bioenergy production and those things. But I think at a broader reach, the biggest thing right now in ag is trying to figure out how to get plants to survive a lot of extreme stress. And over in the past, we've really thought about mineral stress, and growing up in the Midwest, we know a lot about adding fertilizer. And so those fertilizers are usually nitrogen, phosphorus, calcium, those things, and potassium. That was it, not potassium. And so it's always just been replenishing that in the soil, but we've started to notice that the plants still are showing what looks like a nitrogen deficiency. It's not a nitrogen deficiency. The amount of sulfur in the soil itself is depleting itself. And this is probably because the Industrial Revolution kind of realized putting a bunch of sulfur into the air is probably not the greatest thing for everyone. But that really depleted the amount of sulfur going back into our soils. So we're starting to realize that this might be an important element for our plant development. So it kind of would tie back to biofuels eventually, but overall, big picture, it's really important for plant health. The Industrial Revolution, I don't honestly know what years it was, but that was when we were building all these big plants, making a bunch of cars, having a lot of coal burning into the air. So it's a lot of the sulfur that was deposited from that era is in the Northeast, and in the Ohio, Indiana region. obviously the soil we use for a lot of farming and cropping is in the Midwest, so we've started to realize those things now. But the reason why I'm interested in that deficiency in the soil per se, I'm not looking at the soil, but since my genes are sulfate transporters, they're moving sulfur from one place to another throughout the plant. I want to know like how the plant uses that sulfate transporter to move the sulfate, I guess, from the soil throughout the plant.
So I think you already mentioned your project about lipid, the main project about sulfate. I heard you also have a project about orphan gene. Can you tell us something about that?
Yeah, so I don't know a ton about orphan genes. I'm super excited about them. So my orphan genes are located within the xylem structure, and that's kind of how I've adopted them. Since my main project is in those sulfate transporters, which are expressed in the xylem, so I've just been working on these orphan genes, and I'm not really sure what they do. I can't really blast them with enough have a ton of literature because they're only really present in the populus genus. And I have some theory that since they seem to be showing a lot of activity in the xylem, that they have some sort of stress response specifically to this tree species. So maybe they prevent cavitation or like embolism of the xylem in a stress response like this extreme drought or freezing, or they're somehow working with these newfound lipids that are in the xylem structures and working kind of like the core genes found in Arabidopsis that work with MGDG to prevent overall death and freezing. So I think there's some sort of connection between those two because it's a really small protein that is made from these genes. So I think there's some sort of specific stress response to them.
Very interesting. So I think you mentioned their biological function. So do you have any hypothesis about their molecular mechanism?
I have a hypothesis. I haven't explored it a ton yet. Just again because of time. But like I said, there's this gene called the core, I think 15 gene in Arabidopsis, and that mechanism is that that protein folds specifically when it experiences a stress, specifically freezing in that instance, and binds with a lipid in the membrane of plant cells to, I think, make it more rigid. I could be wrong, it might be more fluid, but I think it's rigid in order to prevent an extreme freezing stress from killing that entire cell. So I think the mechanism is likely probably something similar with these orphan genes protein. Just because it is specific in the xylem, I don't think it's maybe binding that way with a lipid. It could be, but since lipids aren't really prevalent in a xylem cell because it's dead, I think it could be working with that lignin structure or maybe working with those xylem rays to get stuff out of the xylem if there is an embolism so that xylem vessel has to shut down. So I think there is some sort of mechanism working there to keep the plant alive during stress response. I'm doing like preliminary salt and drought and mineral stress with them along with my main project. And so some of the findings have been really interesting with them.
Just in case we've got some audience not knowing what is orphan gene, can you explain what is orphan gene a little bit?
Yeah, so an orphan gene, I didn't know what it was when I started this project last year. I was like, that is a new, well, I know that word, but I don't know exactly what it means in this context. So an orphan gene is a gene that's only found within a specific species or within a specific genus. So in our case, I think it is within the populus genus. So overall, all of the poplar trees that we think about in the forest likely has this orphan gene in it. But it's not found in, say, our corn plant. Or at least right now, we don't know if there's a similar gene that lines up with that one. So we do think it's only present in this tree species that we have right now. We haven't sequenced, obviously, a ton of trees species for science. Very few have been. So it could be that it's not an orphan gene, but right now we're considering it specific only to this gene.
In case that we also have some audience not from the plant biology, how important is xylem
Yeah, I do forget sometimes that I speak with other people that don't think about plants in the same way. So xylem is basically the veins of a plant that specific vein moves only the water throughout the plant. So it moves from roots to shoots. And it's really important because plants need water. It's its overall main component of the plant itself. So xylem does move water roots to shoots in one direction, and embolism means that vessel or vein has broken itself, it has ruptured, and it's no longer functional, is no longer able to move water. And that happens in extreme stress events such as drought, heat, and freezing. Those are the main three that causes the xylem vein vessel to rupture. And so that's really a big deal when that happens to a plant because it's shutting off one of its vital life sources of moving water. So being able to either redirect that vein into another one or shutting that down, maybe if it gets infected, is really important for the plant's overall survival and health. And if too many of them rupture, too many of them vessels break, it can ultimately lead the plant to die. Okay, so some preliminary data that I have with the orphans. Originally, we were thinking they were growing a lot faster. So I guess I should actually preface that my orphan plants have these genes knocked out. So these orphan genes are no longer present in the plant itself. So we use CRISPR to mutate it, and so that gene is no longer functional. It doesn't make the protein that it's supposed to for these orphan genes. So the plant itself doesn't have them anymore. So that's probably really important to know. So I've been doing a lot of tests with them along with my sulfur transport trees, and I've started to notice that maybe they don't grow quite as fast per se, but these orphan mutants seem to do a lot better than maybe my wild type, so the trees that are supposed to be quote unquote normal, and my sulfur beans. And they grow either taller, their roots are more established quicker, and they seem to respond to stress a lot better than my wild type plants do. So actually, I have a preliminary trial going right now, and in a media without sulfur at all, they are growing fine. It's grown maybe a centimeter or two more, made a whole new leaf a lot faster than my other plants have. So maybe my original hypothesis is wrong, where those are really important for development in the plant, because it seems without them, they're doing really, really well without the orphan genes. They also do seem to make a lot of anthocyanins, so the color that makes blueberries blue. In their root structures when they're really stressed in comparison to my other mutants and wild type plants. So I'm not sure how that really plays into the plants and what the orphan genes do, but it does seem my plants are doing a lot better without those genes. So I don't know if it's left over from something else and the plant hasn't gotten rid of it.
Would you please remind us again how did you select these two as your target?
Yes. So this was a project that I adopted when joining the lab. So I wasn't part of the original picking of these genes, but the Sci Lab has a huge project working on lignification and wood production in plants. So when they were looking at the 4CL1 gene, which is really important for lignin production, the thing that makes a tree woody in poplar, they found out that these genes were really highly upregulated, meaning they have a lot of expression. This gene is working really hard in the xylem structure itself. So they originally picked these genes. They're like, well, they're important probably in lignin production. They're important in wood production or some sort of stress response, because they did drought and bending experiments as well, and they were still really highly expressed. They were one of the top, I don't know the top, maybe, I'm gonna say 50 because I wanna say 10, but I don't know if it is 10. But it was one of the highest expressed, both of them were one of the high expressed genes in the entire plant itself. So it was really blown out of proportion with the other genes. So that's what led them to choose to pick it, because they're like, wow, that is a lot of gene expression happening in the xylem. So obviously there is some importance to it in lignin production and growth. So it is very confusing when you see a plant without them doing really well, but
Exactly. That's a fascination of biology, I would say.